I don't think aging is one thing. I think aging is multiple different things, and therefore I think it's highly likely that there are different mechanisms that are driving the evolution of this phenomena. So actually my own pet favorite theory for what drives aging is that I'm a big fan of this entropy model of aging. And basically what this relates to is this idea that a cell or a system goes from an ordered to a disordered state. And we believe that one of the major drivers of this is cell division.
Welcome to EHA Unplugged, the official podcast channel of the European Hematology Association, EHA. I'm your host, Isabel Olivera. Today we are joined by Dr. Michael Milsom. Dr. Milsom is a division head at the German Cancer Research Center and group leader at the Heidelberg Institute for Stem Cell Technology and Experimental Medicine in Germany. Dr. Milsom is one of the organizers of EHA's research conference in 2026, Recon 2026, and as an appetizer today, we are going to have a discussion about hematopoiesis and how it changes during aging. Thank you for being with us today.
Thank you for having me.
So to start off, what do you mean by the traditional HSC-centric view of hematopoietic aging? Why do you think everything is centered in HSC?
Well, when we think about how the hematopoietic system changes and evolves during the process of aging, it's a really attractive concept that the hematopoietic stem cells, these cells that we think last throughout your lifetime and are essential for the ongoing production of blood cells and responding to stress when you get depletion of mature cells, that these alterations that would be permanent and progressive should be enacted at the level of hematopoietic stem cells. And there are various kind of conditions where we know that the hematopoietic stem cells, their functionality is altered, often detrimentally during aging. So it kind of makes sense logically that these permanent changes would be as a result of the stem cells getting functionally compromised during the aging process.
What is the main evidence that supports this HSC-centric view?
So I think that there are many lines of evidence that show that hematopoietic stem cells lose their functional potency during the process of aging, both in humans and in experimental model systems like the mouse. So for example, in humans, if you think back to criteria where we look to employ in terms of looking at donors for stem cell transplant, we're typically not using older donors in that setting because there's a higher proportion of graft failure in that setting. And that's thought to result from this loss of functional potency of the hematopoietic stem cells. You can see this really nicely in the mouse setting as well, where paradoxically, the number of immunophenotypic stem cells increases with time. However, the number of functional stem cells declines with time. There are also other lines of evidence, and I think we'll talk a little bit later about clonal hematopoiesis, but I think that this is another kind of excellent line of evidence that something happens at the stem cells level where there's a clonal contraction in the stem cell pool that now impacts on blood cell production and the evolution of a wide range of age-associated diseases. So I think there's lots of evidence that the stem cells alter with aging. But where I think there's a big gap in knowledge is exactly how that might impact on a lot of the clinically relevant features of aging that predispose towards disease. So, for example, immune dysfunction. Is that really driven by altered biology of the hematopoietic stem cells or other cell types? Anemia of aging. Is that really driven by alterations in the stem cells, progenitor cell compartment, or something further downstream? And I think that that's something that's very difficult to address mechanistically, but I think it's a really important question. But I think up until now, we focused a lot on the hematopoietic stem cell compartment.
So you argue that this view is too narrow. Mm-hmm. To focus on HSCs is too narrow. What are we missing if we focus only on HSCs? What else is there?
Okay, so I think that before I jump into why we should look outside HSCs, the hematopoietic stem cell compartment, it's probably important to kind of take a step back and think about broadly what is aging and what does that look like in different individuals. And oftentimes the way that we study this, we'll kind of use something like an experimental mouse cohort, young and aged, and basically look at the things that change on average in those populations in terms of the composition of the blood, the bone marrow, the functionality. And clearly one of the problems with that is that aging isn't just a kind of a process where you go from A to B directly. I think that actually aging is going to multiple different destinations and potentially also at different rates. So if you think about humans, not everybody looks the same in terms of how they elicit age-associated phenotypes. And not everybody develops exactly the same kind of malignant or non-malignant age-associated hematopoietic pathologies. And people develop those at different time points. I think historically we've written that off as alterations in the environment or genetic predisposition. But I can tell you, in our experimental mice that are inbred, our mice look very different in terms of their hematopoietic composition at old age. So I think that it's kind of problematic to think of aging as a single thing. Like we, for example, think about acute myeloid leukemia. In reality, that's a spectrum of different diseases with different drivers that have different aggressiveness. And I think aging is the same. Now, if we kind of pivot back in a very long-winded way back to the question about hematopoietic stem cells and what we're missing, I think that if we just focus in on that population, we probably miss a lot of important information. So I think it's possible that a lot of the phenotypes that develop during aging aren't necessarily as a result of the hematopoietic stem cells being altered. So if our focus is predominantly on the stem cells, we miss that and we're looking in the wrong place for a mechanism that's driving the phenotype. Now, I think there are certain things that are driven predominantly by hematopoietic stem cell dysfunction. So I think in a range of different hematologic malignancies, it's quite clear that a hematopoietic stem cell was likely the cell of origin that became dysfunctional and then later on evolved into a full-blown leukemia. So I'm kind of proposing a more holistic view where we maybe take a step back, don't just focus on one cell compartment, and actually, even if we're interested in hematopoietic stem cells, try to really assess whether in the more mature cell types, or for example, in the niche, whether this is the thing that's really driving the disease phenotype.
So do more mature immune cells show aging phenotypes and that are independent of age?
Yeah, I think that there are a number of clear examples of that. So if you think about defects in the adaptive immune system that evolve during aging, these are a major cause of a range of age-associated predispositions to developing disease. So if you look at, for example, why people generally have a poor response to viral infection, like we saw during the COVID pandemic, or how people respond really poorly to influenza infection, these are generally elicited through defects in T-cell production and potentially also B-cell production and functionality. And I think it's highly likely that the defects there are not related to hematopoietic stem cells. I think that also if we think about some age-associated hematologic malignancies that relate more to the lymphoid branch of hematopoiesis, they're the likely cell of origin probably isn't a hematopoietic stem cell. So these are kind of examples of things that have evolved independent of the hematopoietic system. So actually we've been carrying out a number of studies in the laboratory mouse where it's relatively straightforward to measure multiple different hematopoietic metrics from the same mouse. So, for example, we would transplant hematopoietic stem cells, but also in the same individual, look at anemia, look at the level of peripheral blood cells, look at the gene expression programs, the different cell types. And the amazing thing to me is that actually we see huge diversity there, but we see a very poor correlation between stem cell dysfunction in that individual as measured by what the stem cell does when we transplant it into a recipient and the actual hematopoietic phenotypes in the primary animal. There doesn't seem to be a good correlation which would be predicted in the literature. And in fact, after we've transplanted the stem cells and these cells have regenerated hematopoiesis in the recipient animal, we see a poor correlation in blood cell parameters between the donor and the recipient, which you wouldn't predict if the stem cells were actually driving what was happening in the primary mouse.
Why is there this difference? What else is driving aging in these mice?
Yeah, so this is the million-dollar question. What's the mechanistic driver? So again, I'd revert back to one of my original comments that I don't think aging is one thing. I think aging is multiple different things. And therefore, I think it's highly likely that there are different mechanisms that are driving the evolution of this phenomena. So we can see alterations in niche composition. This is also something that we've studied and found a kind of lack of correlation between this and what's happening in terms of the blood cell compartmentalization. And actually, at the moment, we're really struggling to find anything that correlates particularly well with the phenotypes that we're observing in the mice, even when we look at things that have been previously published in the literature. So actually, my own pet favorite theory for what drives aging is that I'm a big fan of this entropy model of aging. And basically what this relates to is this idea that a cell or a system goes from an ordered to a disordered state. And we believe that one of the major drivers of this is cell division. So, for example, on a very basic level, we know that when you replicate your genome, it's very challenging to replicate it perfectly. I think the same thing holds true, for example, when we try to replicate the epigenome status of a daughter cell compared to the parent cell. So you end up with a diversification. And the more cell divisions, the more you differ from the original cell. So this, we think, is potentially a mechanism through which hematopoietic stem cells achieve diversification. And this diversification can also drive an evolutionary process. So this kind of creates a system of clonal competition, outgrowth of clones with, for example, favorable growth characteristics, and then potentially also disordered functionality afterwards. So this is attractive to me, but that's extremely difficult to study because most of our computational analysis methods tend to focus on what's coherently changing rather than to what degree do we see diversification.
So your work has shown that inflammatory stress, even early in life, can accelerate HSC aging. What can you tell us about this?
Yeah, so we were interested in inflammation and aging for quite a long period of time. So actually, I used to work in the field of inherited bone marrow failures, specifically looking at Fanconi anemia. And it was very clear from the literature that there was this link between inflammation and bone marrow failure in that setting. And so this kind of piqued my interest, and I was working on this in the setting of Fanconi anemia, and it really was kind of one of these eureka moments when I started working in Heidelberg and saw the work of Andreas Trump, who showed that these pro-inflammatory cytokines can push stem cells from this long-term quiescent or dormant state into active cell cycle. So now this idea that cell division is something that drives loss of functionality, it kind of made sense to me that potentially the inflammation was making the stem cells divide. And the more inflammation you would be exposed to, the more those cells would diversify away from the original cells that you'd got in there. So in that context, we started to carry out studies. We had some very elegant mouse models that have been developed by Andreas Trump and her Marika Essers, a former postdoc in his laboratory, is now also a group leader and works in the next office to me. And these mouse models allowed you in vivo to pull apart the stem cells that didn't divide so much with the passage of time. We can even see dormant cells that barely divide during the lifetime of a mouse. And actually, interestingly, these cells retain a young phenotype. And then the stem cells that have divided more. And now you can expose to inflammation. And the interesting thing is that even though all these stem cells are exposed to inflammation, it's the ones that divide that lose the functional potency. And the ones that can still stay dormant, they still retain their functional potency. So importantly, this kind of brings into question this concept of self-renewal, this idea that a stem cell can make a perfect stem cell daughter cell. And I think one of the take-home messages from our work is stem cells can divide to make more stem cells, but very, very rarely do those stem cells have the same functional potency as the parent. So you end up in a situation where each round of inflammation draws some of the stem cell pool into cycle, it loses functional potency, and it never gets regained by those cells. So it's irreversible. And what that means is that now, later on, week, a month, a year later, if you have another stimulus that draws more of the stem cell pool into cycle and they lose their functional potency, you can now have an additive or cumulative depletion of the stem cell pool. And this, to me, is a really attractive mechanism to kind of explain clonal succession with aging, but also clonal contraction during aging.
But this is happening in the HSC cells. So it's been driven by the environment or by inflammation. How much of this increased vulnerability comes from the...
Yeah, so what I was talking about there was really with a focus on the hematopoietic stem cell compartment. So trying to enumerate the number of hematopoietic stem cells. Actually, in these mice that we've exposed to multiple rounds of inflammation, where we've depleted the stem cell pool, actually hematopoiesis looks relatively normal in that setting. And I think that's also what you would predict based on what we know of how things work in humans. Yeah, so we are exposed to multiple rounds of infection, sometimes pretty severe infection. And we always kind of bounce back to these levels where we're able to maintain homeostatic production of blood cells. And interestingly, there are studies of very old humans, so centenarians, who've had a massive contraction of their stem cell pool. Maybe they've got 1 or 2 stem cells that are supporting hematopoiesis, but they're still able to maintain normal levels of peripheral blood cells. And I think that's kind of an important concept, because although the stem cell pool contracts, we still have a system that has evolved in order to be able to sustain blood production during life. So these mice, generally their blood cell systems look pretty normal. I think that what's interesting is when you start to dial in things like these mutations that you see in clonal hematopoiesis. So, for example, the group of Grant Challen and Catherine King have done very elegant studies looking at DNMT3A loss of function and then adding on top of that bacterial infection. And you can see this expansion of the mutant clones in that particular setting. In the mice, I don't think that there's a strong phenotype elicited at the level of the mature blood cells yet. And maybe that relates to, missing ingredients in terms of our mouse models that are important in humans. So most of the time our mice are kept in specified pathogen-free conditions where they're not readily challenged by pathogens. Maybe if we depleted the stem cell pool sufficiently, then added on top some kind of infection, perhaps those mice would have a shorter lifespan or not be able to respond so much to those challenges. But yeah, in that setting, again, we've been very much focused on the stem cell pool rather than any age-associated phenotypes in terms of mature blood cells.
You mentioned clonal hematopoiesis. How does inflammation influence the clonal expansion?
Yeah. Again, I think that's a really interesting and important question. I think that we're only now starting to delve into that to some extent. So I mentioned before the work of people like Grant Challen and Catherine King. I think there's also been very exciting data recently from the likes of Parash Vyas and John Dick looking at human samples where indeed it does look like exposures to inflammatory stimuli, maybe infectious stimuli, provide a selection advantage for example, DNMT3A mutant clones. So I think it isn't 100% clear why that is the case, but I think data does seem to indicate that with each exposure to inflammation and infection, you program the epigenome somewhat along the lines of trained immunity, where if you execute a specific transcriptional program over and over again, there's a residual memory of this in the stem cells. And I guess the idea is that if you now go in again with the same kind of stimulus, like an infection, that the cells will be able to more rapidly respond, or be able to respond with a greater magnitude effect to that same stimulus or something similar. But perhaps there's a little bit of a cost towards this, because hematopoietic stem cells have to be good all-rounders that are able to produce multiple different cell types and carry out different functions. So maybe if you program them in a particular direction, then maybe they don't perform so well as a stem cell. Now, if you look at the DNMT3A setting, I think what's clear is that now these cells have less of this priming effect when you give the inflammatory stimulus. So they have less of an epigenetic memory of that exposure. And it's tempting to speculate that that might actually facilitate these cells still maintaining their stem cell attributes and maybe being able to be resistant to this inflammatory challenge. So I think it's a little bit different than the idea that, for example, a stem cell would get a pro-proliferative mutation, and now this mutation would give a positive selection advantage and allow it to expand more rapidly than the rest of the stem cell pool, but rather this idea that there's a constant ongoing attrition of the stem cell compartment, and maybe things like the DNMT3A mutations, TET2 mutations might make the cells more resistant to that, so they're fitter in that context.
So do you think any of these features can be targeted for delaying or mitigating aging?
Yeah, potentially. I mean, I think that I'm not a big fan of this idea of rejuvenation. I mean, it would be nice, yeah, but I think it's kind of hard to think about exactly how that would work. So even if you look in these extreme settings where people formally demonstrate this, so I'm talking about things like if you put reprogramming factors into cells that you've taken from an elderly organism, or actually if you generate a transgenic mouse where you can transiently induce the reprogramming factors, and now you partially reset the epigenome, and the mice, by and large, look younger and actually have improved health metrics. So first of all, it's obviously very difficult to think about exactly how you're going to facilitate that in the setting of a human. But even when we start to think about fundamental mechanisms that are taking place there, I don't think you're completely resetting the clock on the changes that occur during aging. So obvious example, the somatic mutations that have been acquired, they're still going to be there. So I'd refer back now to the comment that I made towards the beginning of the podcast, where I was talking about aging probably being multiple different things and multiple different destinations. Now, if you ask me, do I think you can correct some of these clinically relevant phenotypes? The answer to that will be yes. And I think that it's not going to be some kind of panacea for all aging phenotypes, but maybe in a more targeted fashion, we could think about addressing things like immune dysfunction, so compromised adaptive immune system. There are some of these pan-hematopoietic rejuvenation strategies that have been put out there, so things like dietary restriction would be one of them, mTOR inhibition, this idea of depleting myeloid-biased aged stem cells. These are all things that have been put forward. I think it'll be interesting to see how robustly those kind of findings can be recapitulated across broad different animal models. I think that there are clearly benefits to things like dietary restriction, but rejuvenation, I'm not so sure that that's a reality. But I would be very happy to be proved wrong.
Is there anything else that you would like to add for the audience?
So I think that one of the things that's important, if you want to do research in this area, is to really kind of take notice of the literature that's out there, but really kind of take a step back and think about broadly, if we're looking at aging, if we kind of think about what we know, what's obvious about kind of human aging, that people develop very kind of vastly different phenotypes at different rates. That's kind of an important thing, I think. And to purely study aging in the context of old versus young, with this idea that we'll be able to identify mechanisms that define what happens to every individual, that seems kind of unlikely. Maybe we need to carry out our analyses and design our experiment in slightly different ways to try and somehow understand this diversification process. Because I think if we can try to understand the basic mechanism that underlies this collapse of the system, then we can actually do meaningful research to think about whether it is possible to delay that or reverse some of the features, or whether that doesn't make any sense at all. And we need to think about things like cell replacement therapies as an opportunity to keep the system functioning for as long as we need it to.
So if I understand well, there is this diversity that we knew already. We know that, during a lifetime, very different things happen, one person compared to another, and that causes aging. But you're saying that there's also this huge diversity in the internal molecular processes in the cells. So the diversity comes from everywhere in the aging process, not only from outside of the body, but also from our biological processes. They're different from one person to the other. So it's really hard to tackle the process.
Although, one of the things that we often kind of debate within the group is we're spending a lot of time and effort and trying to characterize this stochasticity in terms of the diversification of the function of the hematopoietic system. And there, I think the really exciting thing is not just kind of characterize the level of an entire system, but be able to dial into individual cells and clonal systems that occur within the hematopoietic compartment to try to understand what's happening there. So yes, there is this kind of really complex pattern of diversification, but the debate that we have a lot in the laboratory is whether some kind of extrinsic pressure can actually drive you towards a more coherent system, or whether it will actually just, as you were saying, make the diversification even broader. So I think that this is kind of next set of experiments that we'd like to approach, but the problem is studying aging. It takes a long time.
Well, thank you very much for this fascinating conversation. Thank you for being with us today.
Thank you. I've enjoyed it.
Thank you to the audience for listening. If you enjoyed this episode, please like it and subscribe to our channel. Stay tuned for more episodes of EHA Unplugged.